Hi, my name is Alison Fanton. I am a scientist in Patrick Sue's lab at the Arc Institute, and I'm the first author on a recent publication out in Nature Biotechnology called Site-Specific DNA Insertion into the Human Genome with Engineered Recombinases. Genetic engineering has enabled rapid advances in biological research, biotechnology, and therapeutics. We can now investigate genotype-phenotype relationships at a genome-wide scale, produce recombinant proteins and genetically modified crops, and develop therapies to correct diseases at the genetic level. This promise of genetic engineering has driven researchers worldwide to discover and engineer molecular systems capable of making targeted changes to DNA. And to identify these systems, we've turned to nature. Nature has been cutting, copying, pasting, and rearranging DNA for billions of years through the evolutionary arms race between mobile genetic elements and their hosts. In the past few decades, scientists have harnessed these natural mechanisms, including CRISPR systems, transposases, viral vectors, and recombinases, into powerful tools for making precise changes in human cells. Over the last decade, we've really improved our ability to generate precise, small-scale edits, beginning with the discovery of CRISPR programmable nucleases and through the development of sophisticated fusion systems like base and prime editors. But beyond small edits, a hugely important capability is creating multi-kilobase insertions. This enables installing not only entire genes, but also multi-gene cassettes, and the regulatory elements that control gene expression. This capability enables complex synthetic biology like installing circuits and synthetic receptors for gene therapies, and enables the development of drugs to treat genetic diseases with heterogeneous pathological mutations across a patient population. One promising tool for generating precise, large-scale insertions is the large serine recombinase. These proteins mediate recombination between two 40 to 50 base pair DNA attachment sites called attP and attB. These reactions are unidirectional, mediated by a small, around 500 amino acid single effector protein, do not result in double-stranded breaks or rely on host DNA repair pathways, and can accommodate large cargos, making this a potential system for safe and stable integration of large DNA cargos. In previous work from the lab, we conducted a large-scale computational mining effort to identify new orthologs of large serine recombinases. We experimentally characterized their capability to integrate DNA into human cells, focusing on insertion efficiency and genome-wide specificity. This approach uncovered diverse LSR variants from across the microbial world, providing a rich library of enzymes with distinct efficiency and specificity profiles. With these natural systems as beginning scaffolds for engineering, we next sought to develop recombinase systems that robustly and precisely integrate DNA directly into endogenous genomic sequences, rather than requiring a first step of installing an attachment site landing pad. We reasoned that three key mechanistic limitations were impeding efficient and specific integrations: genome recognition and binding, donor DNA binding, and recombination efficiency. We therefore systematically engineered the LSR DN29 to improve these processes, developing three distinct engineering strategies: directed evolution, Cas9 fusions, and donor optimization, to try to maximize on-target integrations, which in the wild-type system occurs at a specificity of 11%, and minimize or eliminate the other 89% of integration events. First, we developed a bacterial selection system for directed evolution. In this process, we performed cycles of generating mutations, selecting out only functional variants, and shuffling these variants to create multi-mutants. When we screened these variants in human cells, we identified a collection of mutations that enhance either integration, efficiency, or specificity, but not both. By performing many iterative rounds of driver mutation stacking, we generated new recombinase variants with enhancements in both characteristics. As a parallel engineering strategy, we hypothesized that fusion to a DNA binding domain, specifically Cas9, could provide a means to recruit LSRs more effectively to the on-target location, thereby boosting both efficiency and specificity of insertion. In this approach, a single guide RNA is engaged with the genome, recruiting the LSR to the on-target site and resulting in around a threefold increase in efficiency. Alternatively, this approach could be utilized to bind both the genome and the donor with a single guide RNA by modifying the donor plasmid to also contain the guide RNA binding site. This method further boosted efficiency up to nearly 40% with wild-type DN29. A final approach we investigated was improving the donor attachment site sequence. We generated libraries of mutated attachment sites, mutating one half of the attachment site at a time, delivered these libraries with the recombinase into cells, and amplified the integrants. This approach enables identification of what nucleotides and what positions are important for recombination, giving us clues to mapping protein DNA binding. Additionally, we identified a new attachment site called the attP that contains enriched mutations and results in improved recombination activity. Finally, by combining these engineering strategies together, we improved DN29 from 3% efficient and 12% specific to up to 20% efficiency and 60% specificity with the directed evolution variants, and finally up to 50% efficient and 97% specific when including the Cas9 fusions and modified attachment sites. Next, we wanted to explore what research and therapeutic applications these new single locus targeting recombinases could enable. We demonstrated integration in challenging cell types, including non-dividing cells, stem cells, and primary T cells. But just integrating the DNA is only the first part. More importantly, the integrated cargo must be functional and stable. To study this, we engineered human embryonic stem cells and induced pluripotent stem cells using hi-fi DN29 Cas9, installing CRISPRi machinery. We then generated stable cell lines through selection and transduced guide RNAs to test the CRISPRi knockdown efficiency. Finally, we differentiated the stem cells into HPCs to test whether transgene expression was still robust in differentiated lineages. We found that these engineered stem cells and differentiated HPCs demonstrated robust knockdown of cell surface markers at comparable levels to classical safe harbor loci such as AAVS1 and Clibel. A key therapeutic application in T cells is integrating synthetic receptors such as CARs for immunotherapies. We developed protocols for plasmid integration in T cells, delivering CD19 targeting CARs. To functionally test these constructs, we cultured the engineered T cells for 13 days to dilute out all episomal expression, and then performed cell-killing assays with NOM6 target cells. We saw high rates of NOM6 killing even at low effector-to-target cell ratios. Finally, not only does a novel safe harbor need to enable robust transgene expression, but it needs to result in minimal perturbation to the natural transcriptome. We performed RNA-seq of these edited stem cell lines at various loci, and interestingly found that LSR-mediated insertions produced the fewest differentially expressed genes compared to all the Cas9 methods. When we look at these differentially expressed genes, we see that none of them are oncogenes or tumor suppressors for the LSR-edited cells, whereas the same is not true for the Cas9-edited cells. Collectively, these data identify AdH1 as a strong safe harbor in clinically relevant cell types, which result in robust editing, expression, and minimal undesired cell perturbation. Finally, I'd like to thank everyone who contributed to this project. It was a huge effort across many colleagues in the Sioux Lab, as well as collaborators across Stanford, UCSF, the ARC Institute, and the University of Tokyo. Thank you for listening, and please reach out if you have any questions or have interest in collaborating or using these recombinases in your own research.